Welcome to this special lecture for meta-analysis. This is special because it will be a combination of masterclass, explainer, and step-by-step guide. And the idea is that I'm going to introduce very briefly the goals of meta-analysis and tell you how to do it in JASP, but we are going to do the very basics. And then at some point I will say, pay attention because this is what the part of meta-analysis that will be covered in the exam. So let's start by saying that the goal of meta-analysis is to make a synthesis of the knowledge we currently have about some area of research. And basically the data that we are going to use is not something that we are going to collect with participants. The data will be published studies or non-published studies. So by published or non-published studies, so I mean, we use the studies that were published in this area, but sometimes we need to try to find some studies that are not published, so we can ask the authors of published studies whether they have additional data, if they know that someone else is doing research in this area and they may have the data but they haven't published, and that's to try to avoid as much as possible the publication bias. Publication bias is the issue that sometimes only the articles that show a significant effect are published, and those that don't are not published, and that biases the meta-analysis. So the meta-analytic authors need to do the effort of trying to find if there are non-published articles or non-published data that should be included in the meta-analysis. So I'll give you an example of JASP, but before that, this is the website of JASP that explains meta-analysis. It's a really concise article of meta-analysis. It tells you not just how to run it in JASP, but it goes through the ideas behind all the analysis. It's a relatively short article and very extensive in the explaining all the parts of meta-analysis that are important. So, but we are not going to go into the detail that this article goes in this lecture. Okay, so this is JASP, and this is the data that we are going to use. This is the data that is available in the library of JASP. So you can have access to this data in JASP itself. So as you can see, instead of having participants in the data, we've got articles. So here in this column, we've got the author's names, and here the year of the publication. And then we've got two really important columns that we need to run the meta-analysis. One is ES, that stands for effect size. So we need to look at a publication, the effect size that we are interested in. So in this case, this is about the effectiveness of a vaccine, and the effect size is log odds, which is the number of positive cases over the number of negative cases in a group that was vaccinated, and that you've got a probability, and then you do the number of positive cases divided by the number of negative cases in the control group, and then a logarithm is calculated in both of those two numbers, and then a subtraction occurs. So that is the effect size. So basically it tells us how effective is the vaccine. And then we've got the standard error. So the standard error has to do with the variability of the data, and it's related to the sample size. So when a study has a large sample size, then the standard error is lower, and when it's a small sample size, then the standard error is larger. So these are the main things. Then we've got other variables that are sometimes just there to illustrate. So this is, in this case, the number of positive cases in the treatment group and the number of negative cases in the treatment group. And then C-pos is the number of positive cases in the control group and the number of negative cases in the control group. So these are useful, but we are not going to use in the analysis. This one has to do with the location of the study. And this is how participants were allocated to the study: random allocation, alternate, and systematic. And this could be useful because we may have some moderation. We may see that there is, if there is a heterogeneity, that means that the effect sizes vary a lot. Then it may be the case that there is one variable that we have in the studies that may explain the effect. Let's say, for example, it may explain the variability. So, for example, if there is a huge variability, perhaps the ones that are with random allocation are more similar to each other than the ones that have a systematic allocation. So that's the type of analysis that is done. But we are not going to do that in this lecture. So we are going to focus on these two columns: the effect size and the standard error. Okay, so we move here, and we've got a we are going to do classical meta-analysis and Bayesian meta-analysis. Bayesian very, very briefly, and within classical meta-analysis we have two types: fixed effects and random effects. So basically, you've got the variables that you saw in the data, and then you basically effect size, you put ES here, effect size standard error, then it goes there. And then you choose the method. Here is fixed effects, and I'm going to explain that. But if you use the arrow, you can use other methods. And here it goes the study labels. So it will this is not part of the analysis, but it makes the output more elegant. So when you get in the output, you will get the name of the authors instead of just study 1, study 2, study 3, study 4. And as I said, covariates and factors, if we believe that one of these variables may affect the effect size, so in different values of these variables, the effect size will be different, then we use it as covariates if it is a numerical variable, or factors if it is a nominal variable. Okay, so basically the first part of the analysis, when we run the analysis, what we get is this table that first does an omnibus test of the model coefficients. And basically, this is testing whether the effect sizes that, the sample of effect sizes that we have comes from a population in which the effect size is 0. And if the p-value is very small, then we reject that hypothesis that the effect sizes of this sample come from a population of effect sizes in which the effect size is 0. So here we've got the p-value is a very, very low value. Remember, e negative 25 means that we need to move this period. 25 places to the left. So it's a very, very low number. So then we reject that. So basically we say in the population, the effect size is not 0. And then the other is the test of heterogeneity. And basically, if we have a low p-value, that means that the heterogeneity, so that means that, let's say that there is the effect sizes that we obtain come from a population in which there is some effect size. And as we explain in sampling distributions, you obtain sampling distributions. You create a sampling distribution by randomly obtaining values from that population, and you obtain the effect size, and you've got a sampling distribution. So basically the p-value is what's the probability of obtaining the variability of effect sizes that we obtain in this sample of effect sizes, if the effect size in the population is such number. Well, the p-value is very low. That means that it's unlikely very unlikely to obtain such variability of effect sizes that we obtain in our study. So we need to do something different. So basically what that tells us that fixed effects analysis is not appropriate. We can still do a fixed effects analysis, but this will apply to just the sample. So within the samples of studies that we have, the estimation we do, it's correct, but we cannot generalize to the population. So basically here it says that the estimate of the effect size is negative 0.436 and the standard error 0.04. So basically it's showing that the effect size is not 0, but we need to do a random effect size meta-analysis to be able to generalize these results to the population. Now, this is important for the exam, the forest plot. You need to be able to interpret what this forest plot is representing. So basically what we have here is the studies. We've got all the studies represented in each of these lines. So sometimes we've got the actual name of the study. If we didn't use the labels, we will have study 1, study 2, study 3, study 4, etc. This is the effect size on that study. Negative 0.94 in Aronson study, negative 1.67 in Ferguson and Symes study, negative 1.39 in Rosenthal et al. Number 1. Okay, so we've got the effect size for all the studies, and we've got a 95% confidence interval for the effect size. For example, in, let's say, Van de Veer et al., the effect size is negative 1.63 with a confidence interval that goes from negative 2.57 to negative 0.70. And here we've got represented graphically what we've got here numerically. So that's the point estimate, and that's the error bars indicate the effect size. Now this vertical line indicates the value 0. So as you can see, we've got effect sizes, most of them on the negative side. This one is 0, well, 0.01. This one is in the positive side, but most of them are on the negative side. And here is FE is fixed effects model, and this is the overall effect size. This is very important. So doing this analysis, we obtain an overall effect size. And the overall effect size has some variability. So this is the overall effect size is represented with a diamond, and the length of the diamond is the confidence interval. So yeah, it is negative, and the confidence interval is doesn't touch 0, so we can be have some certainty that a degree of certainty that the effect size is negative, and this is the numerically the point estimate is negative 0.44 with a confidence interval of negative 0.52 and negative 0.35. Now, then we go to the next one, because fixed effect size, due to the heterogeneity of the data, then it seems that it's not appropriate, so we need to do a random effect size analysis. So we've got it here in the second one. So what we have here is that what we did is that we changed the method. Instead of having the fixed effects, we have restricted ML. There are other methods, but we are not going to go into the details of that. Okay, so now what we get is not only the estimate of the effect size, that is now negative 0.7452, but we also have an estimate of something called tau, or tau square, that is an estimate of the variability that we observe in the effect sizes. So we've got effect sizes of different size, and so they vary. That's the variability. Now, one part of this variability is explained by random variability. So by sampling from an effect size of the population, we obtain different effect sizes in the samples. But some of that variability is not explained by that. So what the different types of random effect analysis do is to give different weight to the studies. So perhaps some studies require more weight than others, and we are going to do an estimation of the overall effect size that takes into account this weighting. Okay, so we've got the forest plot as well, and another. Sorry, before we go to the final plot, the difference is that the overall effect size now changes. So the RE model, so it's a random effects model, produces a different estimation of the overall effect size. So in this case, it's negative 0.75, and the confidence interval goes from negative 1.11 to negative 0.38. And another important thing for the exam, the funnel plot. So you need to understand what's the funnel plot. The funnel plot is one of the methods we have to estimate whether there is a publication bias. So publication bias is that only studies that are significant, they show a significant effect, are published, and those that don't show a significant effect, they tend not to be published. And well, statisticians came up with an idea of measuring that possibility with the studies that we have. And the rationale is the following. The rationale is that the studies that have small standard error, they should be close to the overall effect size. And studies that have larger standard error, they should be more spread. So that's why we've got this funnel or triangle. And here we've got the standard error of the study, and this is the effect size. And the line shows the estimate of the effect size. So the standard error here is the largest, 0.8, and we expect values to be more spread around symmetrically around the estimate of the effect size and more spread. So if we consider this interval between 0.6 and 0.8, we've got 3 studies, and these 3 studies are within the expected range. Between 0.4 and 0.6, well, one is outside, but the other one is okay. It's not symmetrical though. They should be 1 and 1 or a similar amount of studies on the 2 sides. But we have to look at that as overall, not just as intervals. In this interval from 0.2 and 0.4, we see that the studies become more closer, but we've got 3 studies that are outside the expected area. But in terms of symmetry, these 3 here are almost compensating these 3 here. And then in the interval from 0.0 to 0.2 standard error, we've got all the studies outside the expected area, which it's a bit concerning because they should be closer to the effect size, the overall effect size. So still not conclusive. There are other tools to determine whether there is publication bias. This one is not very conclusive. But this is one of the tools that is used in meta-analysis. I will go to the Bayesian aspect, but I'm going to just explain one issue very briefly. So what we have in the Bayesian meta-analysis is a prior over the models and a prior over the effect sizes. So that's the same as the traditional use of Bayesian model comparison. So regarding the models, you got here the prior, and this is similar to Bayesian regression analysis we did. We have a uniform prior. We give the same probability to all the models that we are going to estimate in this case, in this analysis. And we got a prior probability and a posterior probability. And in this case, there will be a model for a null hypothesis of the fixed effect model, another fixed effect model, but that's the alternative hypothesis. So basically, this is saying that the effect size, overall effect size, is different than 0. And then the same for random, a null hypothesis model and an alternative hypothesis model. And then for the effect size, important for the exam is there is a prior, and the prior here is a Cauchy distribution that is similar to a normal, or more similar to a T distribution, and with the location parameter 0 and scale 0.00707. So that means that it's like a normal distribution in the sense that it's symmetric, and it is the center of that distribution is 0. So values that are closer to 0 are more likely than values away from 0. As we saw in other analyses, what you can do with Bayesian analysis is a combination of different models. So here is an estimation of the effect size based on fixed effects. So here is the overall effect size. We've got random effects overall effect size, which is different. And we've got the tau, which is an estimation of the variability of the effect sizes, not explained by random sampling. But this is the feature that is nice to have in Bayesian analysis, which is a combination of these two. So we combine, we give different weighting to these models based on the Bayes factors, or actually in the posterior probability, this one, and then we based on those weightings, we do an estimation of the effect size. And here is this value. And that's all regarding meta-analysis. Again, the goal of meta-analysis is to provide an overall knowledge of some effect size. What we need to do that, we need to find studies that study what we are interested in. These studies have to have an effect size published and a standard error. Sometimes they don't have that, but we can work it out. So if they have, sometimes if we, mean difference is a traditional effect size, but we use a standardized, which is the Cohen's d, and perhaps in the study they didn't publish that, but if they publish the t-score, the mean difference, standard error, then we can work out the effect size. And so when we have the effect size and the standard error, then we can run a meta-analysis, and we do fixed effects and random effects. Fixed effects, when there is the variability of the effect sizes is not too large, that fixed effects would be appropriate. Otherwise, we need to do random effects. And in the Bayesian, there is a combination of the two. And we've got the forest plot, that is a way of representing the effect sizes visually. And we've got the funnel plot, that is a way of representing studies in order to measure whether there was publication bias. Thank you.